welcome to the Talking Crop podcast. My name is Kathryn Seebruck, and I'm a commercial agriculture educator with University of Illinois Extension, serving Daviess-Stephenson, and Winnebago counties. Talking Crop is a row crop production podcast with episodes occurring every other week during the growing season between May and September. In each episode, I bring on a guest speaker to discuss topics related to their areas of expertise. In today's episode, I'm joined by Nathan Schroeder, Associate Professor of Nematode Anatomy and Biology in the Department of Crop Sciences at University of Illinois Urbana-Champaign. We discuss soybean cyst nematodes, their pest status and control, as well as free soybean cyst nematode testing that is available to Illinois producers. In the episode description, you'll find links to that free SCN testing program, as well as the SCN Coalition website, which is a great resource with tools such as a profit checker and field guides. For the next episode of Talking Crop, I'll be joined by Trent Ford, the Illinois State climatologist with the Illinois State Water Survey. We'll be discussing something that we have had no shortage of this season, which is weather extremes in Illinois. That episode will air on Wednesday, July 8th. And now, please enjoy this episode of Talking Crop, Nematode Presence Control and Testing with Nathan Schroeder. Hello, Nate, and welcome to the Talking Crop podcast. Thank you for being here today and speaking with me. It's good to talk to you.
Great to be here.
So I'm going to have you start things off just by introducing yourself. So if you could give us a little bit of your background and talk about what your research focus is.
Sure. So I'm an associate professor in the Department of Crop Sciences at the University of Illinois Urbana-Champaign. My research focus is nematodes, both plant parasitic as well as what we call free-living nematodes, or non-parasitic nematodes. I actually grew up in New York, but most of my extended family is in Illinois and Iowa, soybean farmers in Iowa. And so I had an interest in agriculture, did a PhD up at Wisconsin, and then worked out at New Jersey at Rutgers University before starting here in 2013. So I've been here for about 13 years now.
And so your focus then, like you said, is on nematodes, and we'll get into the specifics of your research in a little while, but I actually wanted to have you take it back to the basics a little bit and talk about what nematodes are as a whole. So a little bit of history for everybody. Dr. Schroeder actually was my professor of, I think it was intro plant pathology back in the day.
Right.
And I remember kind of being blown away by the concept of nematodes, because I think you said that they're like one of the most abundant animals on Earth. Is that correct?
That's correct. Yeah, good memory.
Yeah, thank you. And it was just really surprising to me because you can't even see them, right? When you think of animals, you don't think of nematodes. And so could you just talk about kind of that and what they are and why and how, I guess, they are a pest of field crops?
Yeah, so they're roundworms. They're typically microscopic, as you said, Kathryn. They're everywhere. So most are not parasitic, most of the species out there, but there are definitely some baddies out there. There are ones that are parasitic on plants. There are ones that are parasitic on animals. So if you have a dog and you give your dog heartworm medicine, that's actually preventing a nematode infection of your dog. But for the crop side, there's thousands of species that are parasitic on crops. In Illinois, there are several dozen that we know cause extensive damage. As you said, most of them are microscopic. And you don't see them. They're living below ground. They're feeding on roots. And a lot of times, even the above ground symptoms, you don't really see much of a issue, and it's not until you start looking at the yield where you notice, oh, there's lower yield than I was expecting. What could that be?
Sure. And you sent me some stats about soybean cyst nematode in particular. I think that's a research focus of yours, and that's due to just its kind of prevalence as a pest in field crops, specifically, obviously, soybeans, right?
Yeah, so soybean cyst nematode is considered the number one pathogen of soybeans in the Midwest. So that's more than any fungus or bacteria or virus. It is found now basically wherever soybeans are grown throughout the Midwest. We don't think it's originally from the U.S. It was first discovered in the 1950s and basically spread over the decades, and now is in every county in Illinois, every county in Iowa, um, most of the soybean producing counties in Wisconsin, Indiana, Ohio. Basically, wherever soybeans are grown, you're going to find soybean cyst nematode.
Even though that's the case, so if they're in every single county in the state here and beyond, it's not necessarily to say that everyone's going to have an issue with soybean cyst nematode, right? Like they're not every field we drive by looks like it clearly has some sort of issue going on because of that. So why is it then, like, why is that the case? Why isn't every field negatively affected by that pest?
Well, that's a great question, and it's great because I don't know the answer. We do know that any field could have it, and we emphasize a lot, and I'll try to emphasize here the importance of testing because it's impossible to just drive by and look at it. Now, I know you studied weeds as a master's student. I always make fun of the weed scientists because they've got it easy. They can just sit in their car and they can say, okay, that's a problematic field.
Yep.
I can't do that. I have to actually get out of the truck. So you can't look at the field above ground and say, oh, yep, there's a problem. We don't always know why a particular field has an issue. Sometimes the nematode made it into that field and there are, let's say, production practices that have allowed the nematode to increase. The nematode genetics, we talk about it as a single species, and it is, but it's genetically very diverse. And so there are populations out there that do better on certain soybean genetics and other populations of soybean cyst nematode that don't do as well on certain soybean genetics. And so that, along with all the other stuff that's going on in the soil, the microbial composition, is likely driving whether some fields get really large increases in SCN and some fields tend to not have much problems. We do a lot of testing or looking at nematodes from farmers' fields, and we do see a lot of fields that come back zeros, but then we also see most fields have it. Some fields have a lot of it. And again, it's really impossible to predict just by looking at the field and saying, oh, yeah, that's a problematic field or not.
That's interesting in both ways, in that you don't really know then what causes it to kind of become an issue in a field.
Yeah, so we know that it spreads primarily by us. So basically anything that can move soil can move it. Mm-hmm. It is an animal, but it doesn't really move very far on its own. It's microscopic. It probably could move a yard over the course of a year on its own power. But anything that moves soil is going to be able to move it. So that can be everything from your combine to the boots that you wear. And so that introduces it into a field. And then the sort of local conditions, we see, correlations with sand content, with pH that can cause slight differences. But again, we don't have a good sense of saying, okay, this is a field where it's, really the population has really exploded, and this is why. Sometimes we do if we know farmers are planting susceptible, but nowadays that's actually pretty uncommon for farmers to use a pure susceptible variety. So it's hard to know in most cases why the population has exploded in certain fields and not others.
As a researcher, is that kind of a frustrating concept that you don't have a nice, clear answer to that?
It's frustrating, but it's also good job security. So there's a lot of unanswered questions that are, to me, really fascinating, and to a farmer are really frustrating.
I can very much understand that. We have a new weed that's popped up here in Stephenson County, and I'm like selfishly very excited about it. But I know from a farmer perspective, it's not exciting.
It's not good.
So I understand that. When it comes to control, is it those resistant varieties that are the main form of control against nematodes?
Yeah, for soybean cyst nematode, at least, and maybe we'll talk about other nematodes later, but for soybean cyst nematode, the two main control strategies are soybean resistance, and rotation. So for resistance, the main form of resistance is this, it's called PI 88788. So it's this plant variety that was used decades ago to introduce resistance into commercial soybean varieties. And that's the main form that's used. That's the main form that's available to soybean growers in Illinois. So when we look at what companies are offering and we look at what the resistant source is, about 90% of the soybean varieties are resistant with resistance from PI 88788. There's only a handful of commercial varieties out there that do not have any form of resistance. Most are coming from this PI 88788. Then there's about 9% or so that come from a different source called Peking. And then there's a couple out there, at least in Illinois, that are from a totally different resistant source. But the vast majority are derived from this PI 88788.
And that one, how does it elicit resistance to the pest?
So it's kind of interesting. So soybean cyst nematode, when it hatches, it initially moves into the plant and it sets up a feeding site. Once it sets up that feeding site, it basically stops moving and it just feeds and feeds and grows in size, eventually becoming that round cyst that's actually, it is visible to the naked eye. Once they set up that feeding site, actually their muscles, the nematode muscles, start to degenerate and they can't move again. And the plant actually will, once they start to sense that the nematode is feeding, the plant will self-destroy that feeding site. And now the nematode can't move anymore. They can't get away. They can't find a new feeding site. And so the nematode starves to death. So it's kind of a unique mechanism for controlling the pest.
And so you mentioned that PI 88788 is very, very common. Is there an issue with how common it is? Like, is there tolerance to that resistance mechanism?
Exactly. So we're seeing more and more of that. And we're seeing more SCN populations that are able to reproduce on PI 88788 forms of resistance. And this actually has been tracked. So this form of resistance has been used now for 2 to 3 decades. And nematologists in Illinois, as well as Iowa, my predecessor, have looked sort of historically and tested SCN populations from across the Midwest from farmers' fields and looked at how well they are able to reproduce. And we're seeing an increased number of field populations out there that are able to reproduce on PI 88788 forms of resistance. Now, it's still better than planting a susceptible plant. So it still has efficacy. I'm not saying that, it doesn't work at all. But what we're seeing is sort of this slow moving train that is about to fall off the tracks. And it's not happening like that. It's happening sort of over the course of years where we're seeing more and more SCN populations better able to reproduce on this form of resistance.
And do researchers know how those populations, those resistant populations, are able to still reproduce under those conditions?
We're, in terms of looking at the genetics of it and understanding the biology of that, we're still trying to figure out. That's an active area of research for many researchers across the U.S. We know that oftentimes the nematodes that do reproduce on it are smaller and they have fewer offspring. So on a susceptible plant, a SCN female might have up to 500 eggs. And on a resistant one, maybe she would be able to reproduce, but only have 50 to 100 eggs. So we still have a lot to learn about that process, how it's able to overcome the resistance.
Interesting. So that's there'll hopefully be answers to that in the future. And then are there efforts underway then to come up with more or different genetics that cause a variety to be resistant to SCN or maybe other problematic nematodes?
Definitely. So I think companies, breeders in general are interested in working on this. I mentioned that there is one other source of resistance that is available. It's called the Peking form of resistance. So it's just basically another soybean variety that has been used and crossed with commercial lines in order to create commercial varieties with resistance. And the Peking variety is available. Again, when we look at what companies are offering, it's about 10% of the total commercial varieties. Now, you being in Northern Illinois, it's actually, there's a good number of Peking-based varieties in that relative maturity. When you move down to Southern Illinois, they have very, very few options. I think last I looked, there was maybe one commercial variety in that relative maturity area that had the Peking form of resistance.
So I wanted to switch gears a little bit and talk about something you've already mentioned, at least briefly, and that is testing for nematodes or soybean cyst nematode in particular, because you have a project with the Illinois Soybean Association regarding soybean cyst nematode testing. So do you want to talk about that really quick?
That's correct. So the program has been going on for the past couple of years. It's funded through ISA, and the program is called Free SCN Testing for Illinois Farmers. And basically the idea goes back to that comment I made that it's impossible to just look at a field and know if you have a problem. You have to test. And because of the changing populations of SCN, we know we're seeing more and more problematic fields. But again, you can't look at it and say, oh, I've got a problematic field. You have to get to really, really high SCN numbers to see any sort of above ground changes. It's basically a very insidious pest in that you'll get yield loss, and above ground, the plants look totally fine mid-season. Mm-hmm. So we want farmers to test. So basically, we have a program, we have a website, and I think you'll be able to put that on your site, Kathryn.
Yes.
So basically, farmers can contact us. We can send them basically sampling protocols, sampling bags. They can send that in to us. We will run the test. We will give them an egg count as well as sort of a rough interpretation of what that might mean. Does it mean, oh, you've got a real problem, or okay, you don't have much of a problem? And some sort of guidance on where to go next. But we really recommend farmers do this, even if, let's say, you do a test and you see that you have a low population. I would recommend going back and maybe another 4 or 5 years and doing a test. So we do hear from farmers periodically. When we encourage them to test, they say, oh, I tested back in 2010 and it wasn't that bad. And okay, well, we've had a lot of presidents since 2010. A lot has changed. Stuff has come into the field, out of the field. And so the populations change. And so we really recommend doing that. Even if you had a test, years ago, do it again just for peace of mind. We're trying to get more of that. We've done over the past couple of years, looking at my number, 2,452 tests since 2023. And of those, 74% of those fields have tested positive. So it is all over. Again, there are some fields where we get 0 counts, but we also tell farmers, because of the nature of how you do the sampling, we're only taking maybe a cup of soil from 10 acres. And so that's a small portion. So you might get a 0 count, and it could be that you just got lucky or unlucky, however you want to think about it.
Yeah.
And if you had tested a little bit over, we would have found some SCN, and it wouldn't have been a 0. So we always stress that a 0 number doesn't mean that you're SCN free. It just means that it was basically below the limit of detection.
And you have said a couple times that it's very difficult to know if you have an SCN issue. And so Do you think that like if a producer has a field, let's say, and they think it's generally like low producing, low yielding, and they might attribute it to like soil type, organic matter, slope, what have you, is there a possibility that it could be that they have that, that insidious pest that's kind of doing its thing underground, causing those lower yields, and it would be worth that producer, sending in a sample with that free testing program?
Absolutely. And, again, I think even if you have a field that you think, oh, the yields aren't bad, I would still say it because, again, you might be losing, let's say, 5-10% of your yield and not really knowing it, because with STN, it doesn't just suddenly appear like some diseases do, where, oh, I never had it before, now I have it. It can build up over time. And so you might be like, oh, well, the yields may not be as good as I would have liked, but they're fine. Well, you could be doing better. And so certainly if you have a field where you think, geez, things aren't as good, maybe you're attributing it to nutrient deficiency. I would highly recommend, just like doing a soil nutrient test for SEN.
Yeah, and there's no harm with the program that you guys are doing because it's free, right? So you're not out anything.
Yeah, I mean, I guess it does take the farmer's time, and so they have to do it. And I mean, nobody really likes soil sampling,
Right.
but yeah, we're trying to basically make it as least painful as possible for the farmers.
Right. Yeah. There's no such thing as a free lunch, as they say.
Yes.
And so when a producer gets the results back and they have, that egg count that you described earlier, okay, so now what? Right. So what do they do once they get that information, depending, of course, on what the information might be?
Correct. So we try to simplify things and break it down into a few different categories of egg counts. So if you have like a really high egg count, we actually recommend, definitely going into a non-host crop. So I mentioned earlier that we have resistance. That's one of the main control strategies. And then the other one is rotation. So soybean cyst nematode has a relatively limited host range. It does not infect corn. It does not infect wheat, any sort of grass. And so if you have a really high number, let's say 15,000, 20,000 eggs per 100 cubic centimeters, we would certainly say go into corn, repeat the test after the end of that corn year. The population should have gone down, and depending on how much it goes down, we might say, well, you might actually want to do a second year of corn. If you've been planting resistant soybeans, we certainly recommend, if they're available, going to a different source of resistance. So it could be you have one of those populations. And in fact, with this testing program, for farmers that have really high egg counts, we'll actually do a subsequent bioassay, greenhouse assay, where we'll take those nematodes from that farmer's field, we'll bring it into the greenhouse, and we'll test it against different sources of resistance, just to make sure. In most cases, we are seeing with those high populations that, yes, that population is able to reproduce on PI 88788 to a certain extent in the greenhouse.
And I was going to ask, you kind of already answered my question, but with that just 1 year of rotation, because, it's corn soy rotation, right? It's pretty common. So you would think if someone's doing a corn soy rotation and they have a soybean cyst nematode issue, that rotation might not do much to reduce that population. So do you find more often that folks have to resort to more of like a continuous corn rotation for a little bit? That's going to be the most effective?
So it's really interesting, and this goes back to the biology of the cyst nematode. So during corn years, the populations will go down. Some of the nematodes will die off. However, one of the cool things, and you might remember this from intro plant pathology, so the nematodes start their life off as eggs. And one of the tricks up their sleeves is that they can actually, as an egg or an animal within that eggshell, they can actually sense their outer environment. And so in years where soybeans are planted, they can actually sense chemicals coming off of those soybean roots and say, okay, it's time to hatch. There's lunch ready.
Yeah.
And if they don't sense that, they have a tendency to just hunker down within their eggshell, which provides sort of a protective covering. And so, yes, planting corn for 1 year will reduce the population, but planting it for 1 year will not eliminate it. It won't bring it down to zero. And actually, there's a lot we don't know about that hatching process, but we'll see sort of diminishing returns. So the first year of corn, you'll get a substantial decrease in SCN. The second year of corn, well, you'll get a decrease, but it's going to be less than the first year.
Sure.
Third year, you'll get another decrease. So the nematode it's sort of an internal bed hedging strategy where it doesn't allow all of its brothers and sisters to hatch at once. And so you would need to go probably a decade of corn to totally eliminate it. And unfortunately, because of its ability to just move with soil, it's so easy to introduce it back into a field. We basically tell farmers, look, once you have it, you're never going to get rid of it. You can't eradicate it from a field. The best you can do is manage it. And so we really encourage farmers to think about it that way as sort of a management issue. The analogy that I kind of give is, cholesterol. So, there are those of us that go to the doctor and you're told, okay, you're predisposed to having high cholesterol for whatever reason. And, you can make changes. You can do things that are going to manage that high cholesterol. You can take statins. You can, not eat as much good food. And yeah, you're going to manage it, but it's always going to be there.
Right.
And because of that, the same sort of thing with SCN. Yeah, you've got it. You can manage it. There are things that you can do, but it's not going to totally go away.
And I have one more question before we switch gears to a couple specific projects you're also working on. And that is, you talked earlier about how the nematodes will move with soil. So have you seen, or has anyone, like researchers, seen any correlation of like no-till farmers? Like, do they have inherently lower levels of nematodes, or at least like less spread of nematodes in comparison to those who don't practice no-till?
Yeah, so you might think, there'd be a good relationship. So I've heard people say, I think, tilling will be better because you're moving the soil around, and so it's more likely that the eggs are going to die. The data doesn't bear that out very well. Tillage doesn't seem to do anything. And with no-till, the data is really mixed. You might think, okay, you're going to get less spread. But again, it's just so easy to move that, even in let's say a no-till field, you're going to have areas where you're going to be moving soil, even if it's going to be less. And because it's just, the nematode is so prevalent across the region. When it was first introduced and first starting to come into Illinois, there was a lot of interest in terms of limiting its spread. It's here, it's everywhere. And so the use of no-till, I have seen a couple of studies where that can reduce the population, and it probably has more to do with increasing good microbes in the soil that are helping to break down the nematode. But again, it's not something where I would say, if you go to no-till, that's a management strategy that's guaranteed to reduce the population.
Gotcha. If we could just shift gears and talk briefly about, like I said, a couple other projects that you're working on. The first one being cover crops and nematodes. And I asked this question to a lot of my guests, and that is if they, depending on their area of expertise, if they're seeing like a shift towards looking at kind of the consequences of different conservation practices as they become more prevalent on, said person's area that they research. And so I would assume the answer for you would be at least a little bit of a yes, right? Because you're looking at, I believe, how, different nematodes might be reacting to different cover crop species, correct?
Yeah, so soybean cyst nematode gets the most press in Illinois, but we have a bunch of other species of plant parasitic nematodes that are here that we know can be damaging. A lot of these tend not to reproduce as fast or as much as SCN, so you don't get the buildup as quickly. But we do see fields with high numbers, and typically these are nematodes that actually have a wide host range. They can infect soybean, they can infect corn. And one of the questions we're asking is, in those fields where you're planting cover crops, planting those cover crops may be a host to some of these other pests. And so by extending the amount of time where you're having a host present, are you creating sort of a green bridge for these nematode pests where, normally during the fallow period between corn harvest and soybean planting or vice versa, those nematodes basically have nothing to feed on. By planting that cover crop, are you potentially giving them food such that they can increase their populations more? And so we're looking at basically a bunch of different plant parasitic nematodes and their reaction. And we're also looking at, we're starting to look more at soybean cyst nematodes, which we know a lot of these cover crops are not hosts for. Although one of them we're looking at is called pennycress, which is a cover crop, but people are also interested in it as an oilseed crop. And that one actually, pennycress, which as a weed scientist, it's also a weed, is actually a host for SCN. So it's one of the few non-soybean hosts that are out there. It's not as good of a host as soybean, but it can maintain, at least in the greenhouse, the SCN population.
Okay. And then is there like a plan to take that information and kind of come up with some sort of like resource for folks? Like I imagine something like, I had mentioned to you previously that the Midwest Cover Crop Council has a field guide and they have a table that's similar, I would say, just kind of more broad information about what cover crop species might be host to different nematodes. Are you envisioning something like that coming out of this research?
Yeah, so we certainly know that for some of these nematodes out there, a lot of the cover crops, things like ryegrass, are hosts. What we don't really know is when these winter cover crops, let's say, are planted in the fall and then allowed to grow, the nematodes are they don't have blood, but they're cold-blooded animals, so their activity responds to temperature. So you plant a cover crop, let's say around harvest time of your cash crop. The nematode only has a few weeks before the temperatures in the soil get so cold that basically they're not doing anything. And then a few weeks, let's say in the spring, before you terminate that cover crop. Is that enough time for the nematode to reproduce and increase its population such that it's going to cause damage to that subsequent cash crop? We're also really interested in the sort of specifics. So there's one group of these plant parasitic nematodes called lesion nematodes. So they're the genus Pratolincus. There's probably worldwide 60 or 70 species. We know of at least 8 species that have been found in Illinois. They probably have differences in their host preferences. We really don't know what those are. So while we know that, things like ryegrass are going to be hosts, we don't know, are they better hosts for this species of lesion nematode than this species of lesion nematode? So there's a lot of unanswered questions there that we're looking at.
Yeah, and those will be good questions to be answered as we kind of see that shift towards that increase in cover cropping. So Nate, I have just a last couple questions for you, shifting gears again, and that's actually looking more towards what you primarily focus on in your lab, which is the fundamental properties of nematode biology. Specifically, you guys look at how soybean cyst nematode feeds.
Yeah, so we do a fair amount of applied work, but we also, as biologists, we're really interested in sort of the nitty gritty of what's going on in the nematode. How is it able to do these things that it does? And so one of the things it obviously has to do is it has to feed. And so we're trying to look at basically the mechanisms of that. So specifically, we're using a tool called electron microscopy. So it's kind of like a microscope that It magnifies things, but it does it much, much higher power than a typical light microscope. So it allows you to see things basically at the nanometer scale. And so from that, we can basically create a very, very detailed map of the nematodes cells and how these cells are basically hooked together. And then from that, we can make predictions in terms of saying, okay, this particular muscle is really important for this aspect of feeding. This particular muscle is important for penetrating into the soybean root. And then the long-term goal with that would be able to create targets that basically target individual cells. So there's a lot of nematodes in the soil. If we do a sample from a typical soybean field, we'll find a bunch of plant parasites, but we also find a bunch of what we call free-living nematodes, nematodes that are feeding on bacteria, feeding on fungi, feeding on other nematodes. And those are actually important for soil health. We don't want to get rid of them. Mm-hmm. So we want to be able to create targets that are going to target just the plant parasites and not all of the good nematodes that are in the soil. And so this is sort of a long-term thinking, looking at what's the fundamental biology of these pests. How can we come up with sort of alternative strategies to control them?
So would it be then that you are kind of looking at those mechanisms to then determine how maybe that, like a soybean plant could maybe resist those species because it's somehow targeting those important mechanisms that you were mentioning?
Right. I could think of multiple ways that you could think about either adjusting the soybean in order to modify its traits to better resist the nematode. Alternatively, one control strategy we didn't talk about for SCN as well as other nematodes are seed treatments. So certainly with soybean cyst nematode, resistance and rotation are the main control strategies. However, over the past decade or so, a lot of companies have been coming out with various seed treatments, including ones that are nematicidal, that will basically prevent either the nematode from infecting and finding the soybean root, killing the nematode itself outright. And so you could think of things that were seed treatment based that would basically target that feeding process to either totally eliminate it or reduce it greatly.
I was wondering, as you were talking about how there's the good nematodes, or the ones that don't really do any harm, at least, and then the ones that we're targeting, is it really, really difficult then, are you guys finding, to come up with the mechanisms that could be targeted in the parasitic ones specifically versus the free-living ones?
I think we still don't know a lot about those mechanisms, but I think, industry, so the industry that are developing things like seed treatments are becoming more aware of the fact that we don't just want to kill everything in the soil. Mm-hmm. And so there are some treatments that have come out. We haven't personally evaluated them, that are advertised as being specific for plant parasitic nematodes. And so I think that will be the key going forward, that we want to sort of recognize the importance of soil health. And I think there's a good and growing recognition on the part of farmers that the soil isn't just, let's say, a substrate where you're putting the plants. It's basically its own living organism. There's microbes, there's fungi in there, and there's also nematodes, and sort of thinking about the ecology of that and what's going on. And so trying to have treatments that are going to be economically effective for the farmer while also maintaining sort of the long-term productivity of that soil will be key going forward when we think about the future of plant parasitic nematode control, as well as disease control. There's a lot of other soil-borne fungi that are out there that are also causing damage to growers' fields.
Well, Nate, thank you again so much for joining me today. I've really enjoyed talking to you about your very important and also really fascinating research.
Thank you. It's been great.
Once again, that was Nathan Schroeder, Associate Professor of Nematode Anatomy and Biology in the Department of Crop Sciences at University of Illinois. Be sure to join me for the next episode of Talking Crop, for which I'll be speaking with Trent Ford, the Illinois State climatologist with the Illinois State Water Survey. That episode will release on July 8th. Until then, thanks for talking crop.